he had seen these things, and the Royal Society organized a contingent of trustworthy people. They were all ministers of the church to go visit him. And so they came back and said, it's real. These things exist. And so then for 170 years, people looked at these things. Nobody ever could do it as well as he did because he was a businessman. He wasn't going to share his trade secret on how to make this perfect lens. And so it wasn't for 170 years or 150 years until people developed lens grinding well enough that they could see what he saw and progress to the stage where they'd seen enough cells that they started to say that cells really are the elementary particles of life. No matter what kind of living creature you have, it has cells. That was the beginning. And in the cells is goo. And they called it protoplasm at that time. We call it cytoplasm now. Okay, so cells are the elementary particles of life. And then finally, there was great debate about the origins of life. How do you make things be alive? And Virchow and colleagues, he did a publication, so he's credited, started to see cells divide. So a cell would grow and divide, reproduce. There you go. You have movement, you have reproduction. Cells only come from other cells. You don't have spontaneous generation of life. And that was a big moment in the philosophy of science. So that's the history. Yes?
Could you say again the maximum magnification that
Van Leeuwenhoek? So they have some of his microscopes preserved in the Royal Society collection, and they magnify to 250x. But they know from the notes and the drawings that he had better lenses that he didn't share with the Royal Society that would magnify 500x.
And you ever figure out how he did it?
Yes, just last year. Last year, the Royal Society lent one of his microscopes to some people that did neutron tomography or something that's non-destructive, and so they could look at the structure of the glass lens in that microscope. And it's a perfect sphere, and it's got a little stub. You can see traces of a little stub on one side. And what he had done was pull out a silica glass rod until it was quite fine, break it, and then he held the tip close to a flame, and it melted into a perfect little globule that was a sphere. And then he trimmed off and polished the stub away and put it in a metal frame. And he wouldn't tell anyone how he did it. And they got furious with him.
Bill, I related the same question, and really super duper microscopes now, the biggest objective is always 100x.
Well,
Why is that?
He could see, I don't know, the 100x, what does that mean?
That's for the lenses, for the objective lens.
Yeah.
You get an increase in magnification with the ocular lens.
Exactly.
So
And you can go up to 1,000
100
because you have
times 10.
compound microscopes. They have multiple lenses in a row, and things have very much improved. His lens would basically make a prism out of light also when it diffracted, or when it refracted the light, it would different wavelengths go to different places. So there were color aberrations in the objects, but he didn't care.
I think you'd have a fair amount of spherical aberration too. Because
Yeah.
if you really at extreme magnifications, his spherical aberration is going
It distorts things on the edges of the field of view.
sort of defocuses the edges.
Yeah. So he didn't have a great microscope, but he had great magnification, way better than anybody else did for 50 years. Okay.
And
he didn't commercialize it with his textiles or so, did he do something?
Oh, he just sold his textiles. And he had the best thread for his textiles of any other businessman.
Here we go.
But he also became, this was his hobby, looking at the animalcules. And he became a great scientist. I mean, he was scientific. I mean, he had a scientific nature. And he wrote over 100 letters to the Royal Society that they preserve and publish. And he was a great scientist. He never admitted that he was, though. All right, so that's the history. And now I'd like to show you. Oh, no, I'm not going to show you. This is my notes. We're going to build that. And I hope it might take more time than we have, but in that case, I'll just put the other notes on there and talk about them. But I found in giving these lectures that if I flash slide after slide, it gives you a more detailed view of what's going on, but it doesn't give you as much time to think and to ask questions. So as I draw, you can think. One of the things you can think about is if I leave something out that's obvious, please tell me, because I'm a little absent-minded. Okay. So here we go, a cell. And this is for this August group. This is just my routine that I always do when I lecture. Okay, so Barry told you a lot about atoms, molecules, working on up to DNA, the information storage system that's coded, decoding to make RNA. And then the RNA gets read. The RNA is the recipe card for a protein, usually. And it gets read by the ribosome, the small subunit, the big subunit. The ribosome, as it chugs along, reading the 3 base pair codons of the recipe, chugs out a protein, which is a series of amino acids that have specific chemical features that cause them to fold and do different things. Basically, I find it easiest to think about proteins as machines. So gears, levers, screws. I'm kind of a Newtonian person, but they're machines. They can do work, accomplish tasks. Otherwise, when you think of proteins as these blobs, you think, well, how can that do anything? They are blobs with beautiful shape, amazing 3-dimensional shape, and very amazing chemistry on their surfaces. And so they can modify other chemicals, they can wiggle, they can walk, they can do work. And they're catalysts, okay? They can catalyze a lot of things. Yes, sir. Okay, so let's start with the membrane of the cell. And this is sort of what Hooke saw when he sectioned those pieces of cork. And Barry told you there's a little dimple there. Barry told you that this membrane is actually a hydrophobic layer. It won't let water go through it. It won't let charged molecules of any sort go through it, except at a very low rate. And so it's basically saran wrap that keeps the water on one side different from the water on the other side. And so there can be solutes on one side and proteins on one side that are really highly concentrated and not concentrated on the other side. And so you can specialize compartments by pumping things that you want on one side to create this environment that can do chemistry that the other side can't. And that's the, one of the great powers that evolution has given cells. Okay, these dimples are going to be places where cells move things out of the cell and take things into the cell. This is going to be exocytosis, and this is going to be endocytosis. And this vocabulary is confounding for teaching, but you can't develop the logic to understand things unless you have words to do it, unless you think totally in pictures. So anyway, I apologize for the vocabulary. I'm minimizing it for you. All right, so what do we do now? So we'll put a label here, cell membrane. All right. And of course, you have fluid that's outside and you have fluid that's inside, and the nature of the fluid is defined by the solutes in it, proteins, ions, sugars, things like that. And so outside and inside matter, and I won't write that down, but that's outside, that's inside. All right, so let's get back to something familiar in here, more familiar. We have the nuclear envelope. So this is the nucleus. I'm drawing the envelope, the double membrane around the nucleus. And these interruptions are the pores that Barry told you about, such that there can be regulated traffic of things in and out of the nucleus. And the membrane of the nucleus is actually a continuation of a membrane system that fills the entire cytoplasm of the cell, called the endoplasmic reticulum. It's one of the most wonderful words in our vocabulary. And we just call it ER because it's got too many syllables, but it's a lovely word. So I'll get to that in a minute. The DNA in the nucleus that has the instructions for making proteins and the instructions for making RNAs that will make proteins. All right, and we're going to take, so the RNA polymerase transcribes the information in DNA to make messenger RNA. And so we'll pretend that it's a linear molecule going through the pore. Actually, it wads up into a little wad to go through the pore.
Just from a mechanical point, what's the magnification in order to see that?
In order to see the whole cell?
No,
Yes.
the DNA.
The DNA.
Yeah.
Well, it depends on if the DNA is in its compacted form or if it's a long, thin strand. If it's a long, thin strand, you can't see it with the light microscope.
Okay.
You can see light refract or diffract around, scatter from it, but you can't actually see the object until you get to the electron microscope.
Yeah, that's okay. And what's the magnification of that?
The electron microscope?
To see that.
Oh, right here, you mean for this?
I wonder whether.
What's
I can't
my
see it.
magnification?
Go to my factory there.
Okay, my magnification is 20,000x.
Okay.
I just made that up.
Oh,
But, it's big. I mean, it's a single, it's a string of atoms that has, it's maybe 10 atoms, well, 10 to 20 atoms wide and goes on forever. And an atom, how big is an atom, Barry?
Well, hydrogen is 1 angstrom.
1 angstrom. And
Ten-
how big is an atom?
10 to the minus
Really
10
small.
meters.
10 to the minus.
10 to the minus 10 meters.
10, yeah.
10 to the minus 10 is really, really tiny.
Right, yeah.
Okay, so the RNA is of the same nature. It's a single strand. DNA is most of the time double strand. And just to kind of help you recognize that this is an RNA, I'm putting 5' on one end. And the chemistry defines one end of an RNA or DNA molecule as 5' and the other end is 3'. And things generally read the strand of RNA or DNA from 5' to 3'. And that doesn't really matter. I'm just using it so that you'll recognize I'm talking about RNA here. Okay, so now that messenger RNA comes out here into the cytoplasm of the cell, and it picks up ribosomes, which Barry described to you last week. And the ribosomes, so here's the 5' end. The ribosomes are going to get on at the 5' end, the small subunit and the large subunit. And they're going to start reading those codons and chugging along, and out of a port in the RNA comes the nascent protein, the growing chain of amino acids. And so it comes out and it has a polarity too. One end has a nitrogen group on it. The other end has a carbon carboxyl group on it. So we call them the amino terminus and the carboxyl terminus, but don't worry about that. I'm not going to tell you which end is which on the protein. So a squiggle that doesn't have a 5' on it is a protein in this drawing. All right, so there's a ribosome. Now here's the ribosome that has proceeded further along. In the decoding process. And so it has gone through multiple codons, stuck a lot more amino acids together. And so it actually has quite a large string of amino acids that are getting ready to form the final protein. And the way that happens, let's label this protein synthesis. And the proteins, because of their amino acids chemistry, fold up into a wad. There are parts of the protein that don't want to be in contact with water, so they find one another. And there are parts that like to be in touch with water. They're slightly charged or strongly charged, and they're on the outside. So the greasy parts that don't like water are on the inside of the protein, and the salty parts are on the outside, the charged parts. Okay, so that folding. Now we're going to go very cartoonish. Folding is going to give you proteins of various natures. I'm drawing you here an enzyme, and that little line is going to, when the enzyme gets activated, it becomes a Pac-Man, and that's its active site, and it's going to do something. You know, it might stick a phosphate group on another protein, or it might bite the chain of amino acids apart, the protease that breaks proteins apart. It might add ubiquitin, or it might just bind to another protein and change its shape so that it can be active. So anyway, this is inactive, this is active. Here's a protein we're going to talk about in a bit. It has an extended shape with the ball on one end. This ball actually can eat energy, adenosine triphosphate, that's the main energy source in the cell, ATP. And it can wiggle, and it can act as a motor and drag things through the cell. And that's the one that Barry talked about. That's one of the proteins that I work on. And then other proteins have multiple subunits, they fold, and then they join together to form more complex machinery. And these proteins, we're off chart. These proteins are soluble. All right. So they're floating around in the fluid, the cytosol of the cell. And diffusion gets them around pretty well. If they're not too big, they can, you know, fill the inner space of the cell going where they need to be. Going around membranes, of course. And those membranes, now I'm going to get back to the endoplasmic reticulum. The endoplasmic reticulum is a network of membranes, and they branch, and they are just about everywhere in the cytoplasm of the ordinary cells that we're dealing with right here. So we'll call this ER, and I'll let you just remember that it's called endoplasmic reticulum.
Sure.
Can you say that? Endoplasmic reticulum? All right, so these are hydrophobic membranes. The core of the membrane is hydrophobic, so it creates a new compartment that actually includes the double membrane of the nucleus. And you can have different chemistry in the lumen of this ER tubule than you have in the cytosol. Okay, so there's your first differentiation of chemistry within the cell. And now, so let's have another piece of ER that comes out, and we're going to make it into a flattened disc. And I'm going to have a bud coming off of it, and then it feeds back into a tubule. And of course, these tubule that joins nuclear envelopes, of course, these ER membranes are coming off the nuclear envelope and coming off one another and they're fusing with one another, just making a network of these tubules and flattened areas. So, okay, where am I going now? What I want to do is I want to get from this idea that you can make soluble proteins with ribosomes and get to the idea that you want to make some proteins that are secreted out of the cell. You want to make some proteins that are embedded in membranes to give the membranes their properties, where they can transport in sugar or amino acids. They can link to things on the outside of the cell to give it structure. They can talk to their neighbors through signaling systems that they have. This is about, so the ER is where you make proteins that are going to do those things. So rather than being soluble, these proteins are going to be across the membrane or inside the lumen of the ER. So here's another mRNA, and we're going to swoop it around. I'll just label this again, ER. Swoop it around over here, and here are ribosome subunits. And they are going to be like this, and here is 5' end of the mRNA. Here, the mRNA has captured the small and large subunits of the ribosome, and it's just going to stick out a tiny little stub of a protein out the bottom. That stub is recognized by a protein called the signal recognition protein. The stub has a certain set of amino acids that say grabbed me by the signal recognition particle. And the signal recognition particle then stops translation. It stops the ribosome until the ribosome gets over to a site on the endoplasmic reticulum that has a pore in it. And so now the growing polypeptide, the growing protein, goes through the pore into the lumen of the ER. And as it chugs toward the other end of the mRNA, that protein grows longer. Okay, so we've gotten across this greasy membrane hydrophobic barrier with this protein. And the proteins then fold in the ER, and they can fold into triangles. That's the joke. They fold into complex shapes, but it's easier to draw them as simple shapes. And so there's a triangle that I want to use to be secreted, and the other one's a membrane.
So that's a water-free environment in the ER?
No,
Inside the lumen?
it has water also.
Has water also, but it's greasier, or?
No,
No?
it is just water. It has different
I
proteins.
see.
It has a lot of sugars inside of here. One of the things that happens when these proteins go in, some of them that have the right amino acid signal on them get sugars added to them. So branched sugar chains, and it alters the property of the protein.
So I assume you're talking about the cell before it divides.
Yes.
And what is the timing of that? How this goes, what you're describing? How long does it take? Very quickly?
The growth, it depends on the cell.
Yeah, well.
Yeah. And
you
so
take the cell.
cells in general, it's called the cell cycle. When you have growth and DNA synthesis and all the other things you have to build up to make it big enough to have two functional daughter cells. And that's, we call it S phase, G1, S, G2. And so that's most of the time in the cell cycle, and about 24 hours for a human cell, an epithelial cell that reproduces.
That's what I meant.
Yeah. And then mitosis takes an hour or half an hour, and it splits into two then. Yes.
How many points of contact are there between the nucleus and the bits of the endoplasmic reticulum?
That's a question I don't have a quantitative answer to. A lot. And these pores that things move in and out of the nucleus, there are thousands of them.
And are all the pores on the ER identical?
That is an interesting question. I don't know. I think that they are identical, but people haven't been able to establish that there aren't different sets that do different things.
So when the first cell gets created, is that an act of God, basically? We don't know.
You are asking me philosophical questions. There are people are fascinated, as they have been for 100,000 years, with that question: is what came first? And there are many religious explanations. The biologist evolutionist explanation is that the first cell, or the first environment that created cells spontaneously, no longer exists, but it did once, and it was mud with clay with a pocket that somehow enabled the formation of polymers of nucleic acids, RNA maybe, that then the RNA would polymerize and then disappear, polymerize, disappear, but then RNA of a particular sequence randomly happened, and it was able to template the polymerization of another strand with it, and it was able to replicate itself. So the origin is probably molecules that can replicate themselves, and then evolution of surrounding controlled environment that help them succeed better than their neighbors. So RNA invented DNA to make it more stable, and then maybe some membranes formed because of the just the physical environment had hydrophobic molecules in it, and on and on. You can fantasize as much as you like.
Thank you.
Yeah. Craig Venter, the guy who first sequenced the human genome, is fascinated by this, and he is trying to reproduce. He and many other people are trying to reproduce the conditions in which a cell could form spontaneously.
Maybe similar to that, what's the most basic primitive cell? There's probably going to be a single cell something that exists today. Algae? I mean,
That's a hard question, because all the cells here have been here for more than 1 billion years, 2 billion years, their ancestors. So which one is the most like the ancestral form?
so no, we're asking.
Single cell. It doesn't have a nuclear membrane. It doesn't have a lot of fancy proteins, but it does have a membrane, and it does have DNA, and it does have RNA, and it does have ribosomes. That's a bacterium. And this description could go on for a year. The next fantasy for how our complicated cells came to be. And it's very interesting. But any more questions? Okay, I'm just gonna, I'm probably not gonna get finished here, but that's all right. So we're trying to get proteins outside the cell on this leg, the secretion pathway. And so this protein I've drawn to have a segment that is greasy in the middle. And when it was being translated, that greasy segment stopped in the membrane. It didn't want to go into the water on the other side of the membrane or on this side of the membrane. So it becomes a transmembrane protein. It has a piece that's in the cytosol of the cell and another piece that's in the lumen of the endoplasmic reticulum. I've drawn it with this cup on the end because I want it to be a receptor that receives, that will bind to another protein that perfectly fits in there, and then makes this transmembrane protein wiggle and do things inside the cell. Okay, so let's move them on through the lumen of the ER. Here, I'll call this the lumen. That's some other labels. Oops. All right. So these are going to move out toward the cell surface in a pathway that we call the secretion pathway. And so a little bleb comes off the endoplasmic reticulum in an area that doesn't have ribosomes on it. So these proteins that want to get out of the cell need to migrate in here, probably by diffusion. And There we go. And then this thing needs to move. And what we're going to do, well, let's say where it's going to move to. It's going to move to a stacked array of membranes. It's called the Golgi apparatus. And then it's named after a man who developed a stain, a chemical stain that would bind specifically to it and show it in cells when you're looking at it with a light microscope. And it's called the Golgi stain. He was one of the many contributors to what we know now. Oops. Okay, so this is going to bud off and it's going to become an independent little compartment. And this is too big to diffuse very well. So it uses my protein, or one of my proteins. I mean, the proteins that I study, I don't have any proprietary interest in them.
You didn't invent them?
I did not invent them.
Or secrete them?
I do secrete them. I do. Actually, this protein is not a secreted protein, but so first I have to draw another structure, another concept, which is that inside the cell is just, it's a layer of oil, basically, you know, and that doesn't have much strength. And it's floppy and gooey inside. So how does it have shape? And it has bones. It has bones and it has muscles inside of it that give it shape. And that's called, remarkably, the cytoskeleton, right? And the cytoskeleton, in this case, there's different filaments in it. I mean, they're proteins that polymerize and stick end to end like this and make a long filament. And some proteins, some of those polymers are linear, single folded protein subunits that go like this. They're kind of floppy. Those are called actin filaments. That's what makes most of the protein in your muscle cells. Actin filaments are like ropes. They're good for pulling on things. They're not as good for pushing on things. There's another one that actually is subunits of the same sort, not actin, they're called tubulin, and they will form a hollow tube. And those of you who are physically inclined know that a tube is stiff. It can't bend very easily. And so those are called microtubules. All right, so here near the nucleus, usually, at least in this cell that I'm drawing, there's a fuzzy agglomeration of proteins that we call the centrosome. And the centrosome acts as a organizing center, a nucleation center for microtubules, these stiff polymers. And they can, you know, go out there. Microtubules, the subunits are asymmetric and they polymerize head to tail like this. And so they have a head end and a tail end. And the head end we call the plus end, and the tail end we call the minus end. All right, so there's a microtubule. If you wanted to see it at higher magnification, it would look like this. And these are all of its subunits that give it its properties. Okay, so that's just sort of background. There are many microtubules going throughout the cell, just like the endoplasmic reticulum. All right, so the skeleton is out there, it's a stiffener, and it provides tracks, monorail tracks for motor proteins to drag things along. And so this vesicle is going to use one of those microtubule tracks. And there's its plus end again. It's going to use it to move to the Golgi apparatus. And so here is the motor protein that I showed you down here. And this one is called dynein. And it dimerizes, so there's two subunits. And I guess it's better to use my feet. So there's a long stalk, and there's my feet that have the energy producing site, the catalytic site. Then it changes the shape of the protein so that it kicks forward and binds to the microtubule ahead. And then this one does its thing and lifts off, and then it jiggles around and finds the next binding site forward. And so it walks along the monorail microtubule and drags the vesicle toward the Golgi. And they're machines. Yeah, they are machines. And so the vesicle moves to the inner side of the Golgi, called the cis Golgi, and delivers the proteins to the lumen of this first element of the Golgi stack. And then these stacks, it actually is a conveyor belt. It moves through, and each stack matures into the next stage of the stack, and they shed off the backside, they form on the inner side. That's been controversial how that happened, but people, I think, have finally agreed that things add to this side, this element matures, and then things leave this side.
It's sort of like an escalator.
Yeah, I guess.
Okay, so is it the same thing that went into the Golgi that comes out of the Golgi?
No. Excellent. Excellent. So why are we even doing this stuff? Why is the cell doing this? And it's because, now I told you back here in the lumen of the ER, you can have sugars that get added to the protein. Other things can happen too. You can have proteases that clip off bits of the protein and change its shape. You can have other types of molecules added to the protein that change the charge distribution on its surface and thus change its properties. And so that happens a bit in the lumen of the ER, and it happens more here in the lumen of the Golgi. And we call it processing. And what it really is if you think of it, is here's your initial manufacturing plant. Yeah, you kind of get it set up a little bit and pack it right and get it sent on to the finishing plant. And the finishing plant puts on a layer of nice bright paint, does some scroll work on it, puts some knobs and buttons on it, and then sends it on as a finished product. Processing. Okay, so these proteins go through the processing of the Golgi. And this vesicle buds off. And it's ready to go now. And this one uses, I'm going to make this microtubule go behind the Golgi stack so it doesn't confuse things. And this vesicle is going to use another motor protein, and I'm going to distinguish it by making it have an empty, a white head. Okay, so this one, I didn't label it, did I? Oh no, I'm going to make this one. Yeah, this one I forgot to fill it in. And that protein is called dynein, which has some Latin relationship to motion or force. And this one over here, we're going to call kinesin. The Greek kind is derived from their word for motion. And anyway, so dynein takes it toward microtubule minus ends. Kinesin takes things toward microtubule plus ends. And the vesicle gets up here, fuses, its membrane fuses with the plasma membrane or the cell membrane. And the receptor and the signal, the future signal, get taken out. The receptor is now a transmembrane protein for the cell membrane instead of for all these internal membranes. And the signal is free to go where it wants, or where fluids carry it, or where diffusion. I shouldn't anthropomorphize these things. It's all biased random action driven by physics. Okay, so you have a protein for, we'll call this one for signals. It can also be, I mean, other forms of proteins can be for structure. Cells need to have structure on the outside to give them shape and strength on the outside. So you have, we have, how late do we go? Noon. Okay, we're okay. Structural proteins, collagen. Collagen is one that you've heard of, and it coats the outside out here and gives the cell membrane something to link to out there. Other structural proteins, well, let's just leave it at collagen. Signals, proteins.
Yeah.
For signals and structure, and here is a protein that we want to use as a receptor. All right, and the receptors for what? Now, so they're receptors for signals. Now, presumably, it will be a signal from somewhere else that's coming to talk to this cell. It can be a signal on the neighboring cell that's sticking out on a transmembrane protein from the neighboring cell that touches the receiver protein on our cell and tells the receiver protein to do something different. It gets it to change its behavior, its catalytic behavior. And it can be also free signals that come in and bind. There are receptors for sugars that will help bring sugars inside the cell to produce energy. Receptors for amino acids. There's receptors for little balls of cholesterol that bring cholesterol into the cell. These are very important proteins on the cell surface. There are also, what I'm going to make this into is receptors for, there's the signal, and we're going to call this one growth factor. And the growth factor is going to come in, and it's going to bind to these receptors, and it's going to change them. So the way it's going to change them is it's going to get them to bind to each other. And this signal is a perfect fit for the cup. And it's a perfect electrical charge distribution to match the electrical charge distribution inside the cup. So there's no sloppiness here. A particular signal binds a particular receptor. Sometimes there's more than one type of signal that can bind a receptor, but just leave it as a lock key mechanism. They fit together. And what happens is that in this case, and I'm, semi-inventing this, but this is related to real signaling pathways. The two receptors join together, and having them cluster together gives them the property that they can now bind this protein down here, and it activates that protein. This protein is a catalyst that can now take another component of this signaling pathway, and this one's got 4 subunits that are inactive. Now, this protein is going to break those apart and make them active. And so they become individual active subunits, and they can go on and signal for increased cell growth and cell division. And I've focused on this because next week, Elena Vasky is going to talk to you about cancer. Cancer is all about uncontrolled cell growth and division at inappropriate times and in inappropriate places. There's also elements of the tumorous cells releasing, not caring about their neighbors anymore. They don't care if they're bumping into them, and they don't care to hold on to them so they can float off and become invasive, dangerous cells in your system. Elaine is going to talk in detail about that. But just here's a signaling growth factor receptor, signaling pathway that leads to this stimulating the cell to grow. It's they call it a mitogen because it stimulates mitosis growth. All right. All right, let's turn to another to this. So that's the out. That's the outs. Now the in, cells need to bring in nutrients. They need to bring in a variety of materials. And one of the things they need to do, besides just importing materials that they need as building blocks, is that they bring this signaling complex over and bring it in. And this pathway, I didn't label that, endocytosis. This pathway, in this signaling pathway, you have to turn it off. You don't want the cells to go on dividing and dividing and dividing. And so how do you turn off the signal? And there's a variety of ways to turn off signals. You can change the surface of this this catalyst, this enzyme, so that it closes its mouth. Or another way that it's done is to bring it in by endocytosis into this endosome that then becomes a lysosome. And the lysosome has a bunch of proteases inside of it that have been, those proteases are delivered to the lysosome from the Golgi. They come in, and so it's delivering these proteases. These proteases are not active until this lysosome pumps a bunch of hydrogen ions into it, which makes it really acidic inside, and the acidic environment activates those proteases. And the proteases chew up the receptors. And the growth factor stimulated the mitogenic protein in the signaling pathway. You okay?
Running out of
Running
battery.
He's running out of electrical.
No, I don't need this. I need that.
I got it.
No, it's the same thing.
I'm monitoring the chat.
That means I'm being too long-winded. That should have lasted for the whole lecture. Anyway, this is a way to shut down the signaling pathway and reduce the rate of growth in cell division. If you screw up this pathway, this shutdown pathway, you can have uncontrolled cell growth, which again leads to what we call cancer. Okay. So, oh, that's the wrong place. What did I forget? I think that's kind of the basics of a cell that's in its just, I'm a cell, I'm being a cell, continuing to grow a little bit. Yes.
Going back to where I want to go all the way back to the replicating the RNA messenger and all the ribosomes coming in, tRNA, trying to find the place. Then there are specific things, they're getting a signal for the protein to become, certain parts of the body, and it's very specific. Then it goes up to the treatment, the processing plant, and it gets processed, and you said then it's ready to leave. How specific is it when it's ready to leave, as far as going into the body, what it will become? It looks like there's a lot of changing going on even after it goes out.
There are changes that happen after things get out here because there are enzymes that can work and modify these proteins on the outside as well as on the inside of the cell. You had another element to your question, which was how do you know when it's ready to go, when the manufacturing process is finished? And there are particular sugar residues. In the one pathway that's the best known, sugar residues are put on a protein. In this case, this pathway takes it to the lysosome or the endosome that forms from this, and it gets mannose 6-phosphate sugars stuck on it. And the mannose 6-phosphate triggers the machinery that will make this vesicle bud off here. And it also signals the outside of the vesicle in ways that allow it to fuse only with this membrane. So there's a whole variety of chemistry on the surface of this specific vesicle that says, you don't get to fuse here, you must fuse here. And so even though the vesicle might be moving randomly around the cell and bumping into ER and bumping into other things, the only place it's going to stick and fuse is here. So that mannose 6-phosphate for the endosome delivery pathway is what gives it its specificity. Sure.
So assume this is a human cell, it's a healthy cell, a perfect cell. So what is the trigger mechanism so that it duplicates it?
To make it reproduce?
Yeah.
So this growth factor.
A human embryo is 1 cell, 2 cell, 4 cell. That's what I'm talking
Susan? So the signals that drive the cell cycle in an embryo those are called cyclins. So these are proteins that rise and fall during, so you have, if you have a circular cell cycle, I'm not going to draw this out, a circular cell cycle, how do you drive it around the circle and having the sequence of steps be correct, the chemistry steps? And there are proteins called cyclins that increase in concentration and then disappear. An increase in concentration on a different part of the cycle, a different cyclin, it disappears. So cyclin 1, cyclin 2, cyclin 3, cyclin 4. And so it's like a clock. And you have your cyclin burst, and it does synthesis of DNA. You have another cyclin burst that says grow the cell, which is basically what's happening here. You have another burst and you and then you go into mitosis, which is the chromosomes condense, the nucleus dissolves, you build up microtubules that pull the chromosomes apart, and then you have actin filaments that form a ring that pinch the cell between the two new nuclei to give you the new daughter cells. And so the cyclins in embryos drive the reproductive cycle. And they stop when you get to a cell that should become a neuron and never divide again. Yes.
So, are lysosomes inherently destructive?
The interior of the lysosome is, it's the recycling center. And so it's generally taking things apart to their subunits, amino acids, sugars, lipids. Lipids go through there and get broken down into small fatty acid molecules.
And that's what's extruded, all the.
So, and those come out of the lysosome through various portals.
And then recycle. And
Yeah, and so they get used again.
Okay.
I've always been sort of taught that ATP is the energy currency, and I've always wondered, how is it utilized physically? You talk about movement, or is it reduced and heat is produced, or how is it actually function?
That's a large question. And I can give you a fuzzy answer to it. What happens is you have your Pac-Man enzyme, and ATP goes into that site. Okay, when the ATP is just it's one of the nucleic acids that makes DNA. And it has an adenosine ring, the sugar ring, and then it has phosphate, oxygen, phosphate, oxygen, phosphate, oxygen, phosphate. So TP means triple phosphate. And each of those bonds between the phosphates has energy. A bond has energy. And when you break the bond, you can use that energy to do something. And so that terminal phosphate, we call it the gamma phosphate, when the enzyme breaks that bond, it liberates 9.6 kilocalories of energy per mole. Whatever. Sorry, it liberates energy. And the energy can be used however this enzyme is designed to use it. Now, the protein that I work on, the motor protein, what happens is, let's see now. What am I going to do? It's hard to tell what's my head and my feet and everything, but basically, here's the active site right here. Grab ATP, and when ATP is, so the motor binds to the microtubule monorail, and that opens the site so this can bind the ATP. And when the ATP binds, it locks down harder on the microtubule. And then the ATP, it's because the ATP binding changes the shape of the motor. Just because it's chemistry, it's a new chemical added to the system. And so then ATP is hydrolyzed. And what happens is, if you imagine the vesicle is up there on the ceiling and my arm reaches to the ceiling, okay? What happens when the ATP, when you break that gamma phosphate off of it, that energy is used to open a slit on the side of the motor protein. And then my elbow is it has charges on it that match the charges that were previously hidden in the slit. And it goes slam. And so it pulls the vesicle forward going that way. That's how the ATP works in that case. It changes the shape of the protein in a particular precise fashion such that it moves the vesicle. And then later, okay, the ATP is released, or the ADP is released along with the liberated phosphate, and that loosens things so it can't hold on to its site on the microtubule, and its sibling head over here then picks up an ATP and crunches down on the microtubule here, and so it goes like this down the microtubule. Yes.
What's the mechanism when you lose a telomere?
When you lose a telomere, mechanism. So do you mean for losing it, or what happens after you lose it? For losing it,
Yeah.
you don't. So that's an accident. That's a chemical accident.
That's what.
Yeah, as far as I know, it could. I mean, Susan knows more about this than I do because she talks a lot with Carol Greider, who's our new faculty member who works on telomeres. She discovered the enzyme that puts telomeres on ends. I don't know about enzymes that take that shorten telomeres that take them off. I know that it happens by accident, just because of the way telomeres are formed. They make kind of a hairpin at the end of a double strand DNA to protect the wiggly ends, to close the loop.
So it's just like a statistical.
Well, she presents it as statistical, but not quite. Some telomeres shorten rapidly, more rapidly than others. And how we can stop it from happening, that would be really marketable.
So I'll be Carol Greider for a minute. When DNA is replicated, there's a failure at the end. It just, the DNA polymerase doesn't work. So you need a special machine called telomerase, and it adds little units to keep the telomeres long. And each chromosome end has a certain number of those little telomere units, its own certain number, which is remarkable that I just discovered, and it's had a big impact. If telomerase is defective, then the ends become progressively shorter. And then the ends of the chromosomes are unprotected and they start fusing with each other. And that's really bad. I mean, telomeres are protecting the ends of chromosomes so they don't, chromosome 2, chromosome 7, that very talked about. And you want them to stay separate. But if they don't have their telomeres, then they fuse, and that's bad. So telomeres are the little, did you talk about
the caps on the ends?
The caps on the end of shoelaces.
I didn't. So Bill, I mentioned in the announcement that you work on these motors and work on a connection to neurodegeneration. So why don't you just tell us just a little bit about what you've done with that?
What turns you on there?
So when I started my formal career as a researcher scientist, we didn't know about the motors inside of cells that move things, except for the myosin motor that walks on actin filaments to make your muscles contract. So we knew a lot about that motor from the 1940s onward, people have been doing good biochemistry on that. But we didn't know about the motors that move chromosomes, that move vesicles and other things. So when I was in graduate school, this protein was first isolated. It was isolated from neurons. And people thought, well, maybe this is the thing that helps neurons transport things from the cell body all the way to the end down here to operate this muscle. So a neuron, if I'm the neuron cell body, I'm the manufacturing center, then there's a long extension, the signaling wire that goes to the muscle down there, or it can go to another neuron to signal another neuron. There isn't very much manufacturing capacity down in that, we call it the axon. And so stuff has to be made here and transported out all the way to the end and all the way back to keep that end functioning. Take new stuff out, take worn out parts, bring them back, run them through the recycling machine. And so looking in axons was a good place to look for a motor protein that carries vesicles and other things on down to the tip. And Ron Vail and Mike Sheets discovered kinesin, along with Bob Allen, Scott Gray. They had a competitive relationship. But that was when I was in grad school, so I got fascinated by these motors and how they work. And so what I did as a postdoc was to, I decided we had to figure out what the motors did, what processes they contributed to. We should use genetics, and we should disrupt the gene that codes for that protein, see what happens to the cell. It's a classic approach to trying to understand machinery. You want to understand how your engine works in your car, you start pulling things out, right? Pull off a plug wire, it starts to run rough. You pull off the main distributor cap, it stops completely. And so you learn about things by breaking them. And so I set off as a postdoc in Beth Raff's lab at Indiana University to break the gene for that protein. When we broke it, this was using Drosophila fruit flies, great genetic system. And their proteins and genes are like 85% identical to humans. So almost the same machinery on a cell by cell basis. The neurons are quite different. In a human, neuron goes a meter from here to here and a meter from here down to my toe, assuming I'm still 6 feet tall. So in flies, it's different. But what happened is when we, Beth and I, got the mutants finally, it took about 3 years to get the gene mutated because we didn't have the CRISPR and the other modern tricks that people use now. X-rays and things to break the chromosomes and randomly find the mutation in this gene. And so we found it, and what happened was that the animals died. They no longer survived. And so, okay. But what was interesting was that the larvae, instead of crawling regularly, became paralyzed in their tail end.
It went away.
Long neurons have long axons that go to the tail, and shorter axons that go to the front. And so this protein was isolated from neurons, kinesin. It's present in all cells though, but there's a lot of it in neurons. And we thought, well, that's, I guess, what you would expect if you're having a disruption in the delivery of the machinery parts that you need to operate the whole axon. And it turned out, as it took years, but we worked our way towards finding that yes, when you mess with the motor driven delivery system, you have the axon gets big swellings in it that are all filled with organelles, and they gum up the works. and you don't get delivery, and the tail end of the neuron starts to shrivel up. And it's called dying back. And it's exactly what Lou Gehrig's disease is, ALS, and Charcot-Marie-Tooth disease, things like that. So dying back neuropathy is a classic neurodegeneration phenotype. We found it in flies. And then after that, people got more interested in this protein, who were human geneticists, and they mapped mutations for Charcot-Marie-Tooth disease, and more recently ALS. And mutations in this gene can cause heritable forms of those diseases. And so that was our contribution.
Great.
We help develop a system that can be used to study neurodegenerative disease.
Great.
But I still, and I tried hard, my students tried hard to use genetic screens in Drosophila to find things that would suppress the loss of that gene. Unsuccessful. The system's too complicated for a simple cure.
I have another question. Just some of the AI has done that it will be useful to make it easier to visualize, like folding proteins, considered extremely complicated.
Yeah,
Is that becoming really helpful at this point?
it's real. It's working. It's amazing.
Okay, because it sounded like when you said immense complexity.
Immense complexity.
Right, but this is what it's useful.
Yeah, and so AI, I mean, Barry talked about protein folding, and AI allows us now to predict a lot of protein structures just based on the amino acid sequence. A problem that people have worked on for, I don't know, 40, 50 years, by hand and with computers and, crystallography and diffraction patterns. And finally, just amino acid sequence, a really big computer can do all the different possible folding ways and see which one is the most stable energy form, which side chains of which amino acids get to touch which others, and what makes the protein the most stable. And so that's amazing. In terms of using AI to solve questions about how does a neuron degenerate, I have hope that it's going to happen, but we aren't even close to that yet. Okay, so I have 0 time left. I wanted to talk to you a little bit about your favorite virus. And so I'll just talk my way through it. All right. So endocytosis. So the SARS-CoV-2. How does it work in the cell? This is actually interesting, and preparing for this lecture gave me an excuse to go look it up. what people have learned in the last 3 years. And they haven't really got it nailed down, but they have made really good progress really fast because of all the money that came into it. And what happens is that maybe I can just mess up our diagram a little bit here. Here's the virus, and there's its RNA inside. And the RNA is its genome. It's surrounded by a membrane. So it's the second severe acute respiratory syndrome coronavirus. So they call it COVID-2. Anyway, this thing has the infamous spike proteins on it. That you've read about that change, so that now there's a new strain and now we have to get another shot, and you might get sick again. So spike proteins, and what happens is that there are transmembrane proteins on the cell that are called ACE2. and it's a receptor type protein. They call it the ACE2 receptor. What its normal job is to take a signaling molecule called angiotensin, that's created by ACE1. So ACE1 gets angiotensin activated outside the cell, and ACE2 deactivates it. So ACE2 bites it in half and kills it. Angiotensin. Angiotensin is a signaling molecule that signals distress by a cell. And so your immune system comes in and causes inflammatory processes in the vicinity where there's a lot of angiotensin. So ACE1 creates angiotensin, ACE2 keeps it from getting too concentrated so you don't have inflammation happen. But what angiotensin's positive purpose is probably just to ward off invaders and maybe to get rid of cells that have been invaded. So anyway, the ACE2 receptor turns out to be a perfect match for the virus spike protein. And what happens is that this combination comes in here by endocytosis. There's still some controversy about exactly how this happens. And there's its receptor, and it comes in. And when this, I'm going to make a new endosome down here. And when this endosome acidifies and gets to this point like a lysosome, what happens is that the virus membrane fuses with the lysosome membrane. And what you get, here's the lysosome membrane. And here's the virus membrane. And it's got its spikes on it. And let's say that its receptor now has been degraded. So it's just floating around in the lysosome. What it does, it has some chemistry that allows fusion of its membrane with the lysosome membrane. And the RNA gets out, the viral RNA. And then the viral RNA has accompanying proteins that say, Yahoo, look at all these resources in this cell. Let's make a bunch more viruses. And so, and those are really fascinating molecular biology processes that people are learning a great deal about. And there are targets in there for interrupting the virus reproduction cycle. But that's too much for us and too much for me. So anyway, this process of endocytosis is what gets the virus in. What happens, people think, is that in addition to the virus coming into the cell and starting its replication cycle, taking over all the machinery in the cell, filling it up with virus, the cell breaks, releases all that virus to go to your neighbor cells in your lung epithelia or elsewhere. That's bad. But another thing they think is bad is that the virus, when it's at high concentration, binds up ACE2 and stops ACE2 from functioning. It causes a destruction of the ACE2 receptor in here in the lysosome. So now all of a sudden you have ACE1 cranking away, creating angiotensin. You have high levels of angiotensin buildup, signals to your immune system. Bad stuff going on, come on in here and nuke this place. And so you have tissue damage from the high angiotensin levels.
So what is the vaccine doing with the rMNA?
The vaccine the vaccine brings RNA, let's put it at the bottom here. And Barry talked about this last week. Is the vaccine RNA? It's just a little short messenger type R. It's an RNA that from which messenger RNA can be derived. And I don't know how they deliver it to a cell. Do you guys?
I think it's a little lipid compartment.
So they put some lipids around it. Maybe it's sort of like a detergent. There's some kind of little bubble of hydrophobic stuff that helps it get through the membrane. And then that, there you go, it's an RNA. And this grabs ribosomes and it's translated and you end up making proteins. And I suspect that they are going to go be secreted. And they're secreted, and then your immune cells, the macrophages and other white cells, they pick up those things and say, whoa, what are you doing here? You're not Bill Saxton's protein. You are a foreign invader. I'm going to make an antibody against that invader. And so the immune cells pick it up. They say, whoa, this is bad. Make a whole bunch of strong antibodies. Then those antibodies can come. And since this virus protein codes, this virus makes a protein that is similar to the spike protein or other viral surface proteins. The antibodies can come and bind to the virus. The new antibodies that your body made can come and bind to the virus, and then the other immune cells can come in and eat the virus.
So how does Paxlovid work? Because that's not a vaccine.
Paxlovid, I think, is a protein that stops the replicase, the RNA replicase. Is that right?
Yeah, it's or the processing of that multi-protein that comes off the virus.
Okay.
Effectively, yes.
In the biochemistry of
I think it's a protease inhibitor.
the downstream biochemistry from the release of the viral RNA, the virus RNA needs to replicate itself so it can make more viral genomes, and it needs to produce proteins that are going to wrap up that RNA, and it needs to make proteins for spikes and to gather new. There's a whole bunch of biochemistry that goes to building a new virus. Paxlovid, I think, stops a replicase. Barry thinks maybe it stops assembly of proteins. I'm fuzzy on it.
I think there is some research on drugs that are angiotensin inhibitors. That are
Yes.
used.
Yes, people are looking at that as a pathway to,
It's actually been used in other countries.
but not here because
Right.
we're so cautious.
Where's that money going? Because it's much cheaper.
Anyway, it will suppress it. Thank you, and I'm sorry I ran over.
Oh, we're glad.
Thank you.
All right. And so if you want to hang out and talk, I'm happy to stay here.